This is part 4 of the chapter 2 lecture notes, and we should finish up because we're on slide 34 and we've only got 39. So um, we talked about a few disease-causing bacteria, but it's in prokaryotes. But it's important to know that most are thought to be beneficial. So biological nitrogen fixation (BNF) is exclusively carried out by prokaryotes. So nitrogen fixation, meaning converting atmospheric nitrogen into usable nitrogen, usually ammonium first, which is not usable, ammonium ions, and then converted to nitrites and nitrates, which are, that's a way that plants can get nitrogen and animals can get nitrogen by eating the plants. So, and nitrogen is found in proteins, nitrogen is found in DNA, so nitrogen is needed for sure by living things. Certain examples of nitrogen fixers are soil bacteria, cyanobacteria, which are also photosynthetic, Frankia species, which is a filamentous bacteria that interacts with actinorhizal plants such as alder, bayberry, and sweet fern. And then biological nitrogen fixation is the second most important biological process on Earth after photosynthesis. So that is important to know. And this mostly occurs from bacteria. Bacteria are used to make food. You probably know this, that bacteria are used to make cheese. That's something that anything fermented, but that's something that a lab that sometimes will be done in microbiology classes is making cottage cheese and also down here making yogurt. In fact, you can see this was done in a lab. The students don't typically eat it, but they go through the process of it. And this is a fermented beverage of some sort. Beer? Oh, I'm sorry. Obviously, I just wasn't looking at it right. That's what I'll blame it on. But anyway, then there's wine. So alcoholic beverages, of course, are fermented products. And there's many other examples, soy products and like soy sauce and just anything that goes through the fermentation process uses bacteria. So another benefit from microorganisms is bioremediation, and bioremediation is the use of prokaryotes, archaea and bacteria, to remove pollution. And that has been used, you've probably seen it in the news and seen it in the media to clean up oil spills. But it's been used to remove agricultural chemicals that leach from the soil into the groundwater, pesticides and fertilizers, certain toxins, metals and oxides such as selenium and arsenic can be removed by bioremediation, and the cleanup of oil spills, which I mentioned. The microbiome, microbes help us digest our food. They produce crucial nutrients for us. They protect us from pathogens. They either do it directly by destroying other microbes, or they, just because they live there, they kind of take up the space that other bacteria would have to live. So they're sort of like if they inhabit our skin, then other harmful bacteria can't take their place. There's no actual area for them to infect. So that's why they say, I'm not telling you not to take a bath, but that's why they say you can actually wash yourself too much, with soap. So each individual organism, human, has a normal microbial flora, and it's called the gut microbiota. And you have probably heard about probiotics. That these are in nutrition, you really hear about probiotics and gut health, that probiotics can fix a lot of problems related to gut health, including they even say that if your gut microbiota is off, then is not correct, then or the correct balance, then you can even have obesity problems, you can have inflammation problems, different things like that. So these normal microbial flora or gut microbiota refers to the collective of the collection of bacteria in each person's stomach. And probiotics contain, it's like adding more bacteria into your GI tract. And scientists are also discovering the absence of certain key microbes from our intestinal tract may set us up for a variety of problems, which is what I was just talking about. All right, now we're on the practice question. What impact might antibiotics have on the digestive process? If antibiotics destroy not only disease-causing bacteria, but also our microflora, then that could affect our digestion. We could have some problems because it could affect our digestion of certain foods, and it can also cause us to be open maybe to infection from other bacteria. So we just don't need to overuse antibiotics. The diversity of prokaryotic cells, we covered that pretty well in the notes. The structure, we talked about the plasma membrane, the of archaea versus bacteria. We talked about the cell wall of bacteria is peptidoglycan. We talked about how it could be thick or thin, resulting in gram-positive or gram-negative bacteria. And we talked about the structure of archaea cell walls is not peptidoglycan. Metabolic needs of prokaryotes, make sure you remember the terms photoautotroph, photoheterotroph, chemoautotroph, and chemoheterotroph. Go back and review those if you need to. And then chemoheterotroph. Common bacterial diseases. I didn't name a whole lot, but make sure you go back to the study plan and look. There was a whole lot of examples in the slides when we did viruses. The slides don't have a lot of examples of bacterial diseases in humans. I can name some bacterial diseases like I said E. coli and botulism from the Clostridium botulinum. There's anthrax. There's staph infections. Staphylococcus aureus is a skin infection. There is streptococcus, which causes strep throat. Different species of streptococcus bacteria. There's lots of different ones. The one that I didn't name any that cause STDs, but the one that causes chlamydia, the one that causes gonorrhea, these are bacterial infections, not and syphilis. These are I'm not sure about syphilis, but chlamydia and gonorrhea are bacterial, not viral STDs. And then beneficial prokaryotes, the nitrogen fixing, the ones that can do bioremediation, the decomposers, the ones in our microflora, in our gut bacteria. So that's a good review of the beneficial bacteria. And that'll be it for this